Hi, it's Mr. Evans. Welcome to Room 310 Biology and our fifth HSA review. We'll be covering molecular genetics and evolution, so I wanted to start with the central dogma of biology. DNA, the information storage molecule, DNA carries information needed to make a protein, but it can't make a protein all by itself. In the process of transcription, the information from DNA is copied to a molecule of RNA. RNA can leave the nucleus of a eukaryotic cell, go out to a ribosome, and that's where proteins will be assembled. The information in DNA is used to build proteins. Proteins give an organism its traits, its phenotype. Differences, variations in phenotype, that's what leads to the process of evolution. So let's take a closer look at the structure of DNA. Remember, DNA is a double helix, long chains of nucleotides, and each building block of DNA, each nucleotide is made out of a phosphate group that's attached to a sugar. The sugar happens to be deoxyribose. That's why it's called DNA. The D is the deoxyribose. And then there's a nitrogen base that's attached to that sugar. And there are 4 different nitrogen bases in DNA: adenine, thymine, cytosine, and guanine. If we did some base pairing practice, this is certainly something you're asked to do on the exam. You're probably halfway down this list already. I have one strand of DNA. What's complementary to it on the opposite strand of DNA? This nitrogen base T pairs with an A with a T, C with a G, cytosine with guanine, guanine with cytosine, C with a G, adenine with thymine on the opposite strand. If we look a little bit closer at RNA, you might notice right off the bat, RNA is a single-stranded molecule, not a double helix. RNA is just one strand, but its basic building block, its monomer, is still nucleotides. There's still a phosphate group. The phosphate group is still attached to a sugar, but this time the sugar is ribose. And there's still a nitrogen base hanging off of that sugar. And the nitrogen bases in this case look familiar to us. There's still a C, there's still a G, still an A, but there is no thymine. Instead, there's a uracil. We'll abbreviate that with a U. But once again, this is still a nucleotide, a building block of a nucleic acid, in this case RNA. So we'll practice our base pairing one more time today. And if I'm looking at this strand of DNA, it's complementary DNA. T pairs with A with T, C with G, C with G, G with C, C with a G, and an adenine pairs with a thymine. Well, the rules switch just a little bit with RNA. The T in the DNA is going to pair with an A, but the A in DNA will pair with a uracil in RNA. C with a G, C with a G, G with a C, C with a G, but remember this A is going to pair with a U in RNA. This table here is the codon chart. You may be given one on the exam or a piece of one on the exam. A codon is a group of 3 RNA nucleotides, and it takes a minimum of 3 to code for 1 amino acid. Amino acids are the building blocks of proteins. So for instance, if we were given the combination CGA as a codon, which amino acid is that? First letter C, second letter G, third letter A. Here's my CGA, and that codes for ARG, or arginine. Protein synthesis, if we take a look at the entire process, works this way. The genes are located on the DNA unwinds, and a piece of messenger RNA is built alongside the DNA. That process is called transcription, copying the information from DNA to RNA. The messenger RNA can leave the nucleus, go out in the cytoplasm to a ribosome. The ribosome is where the amino acid chain, which becomes a protein, is assembled. That process is called translation. An easy way to remember the order: transcription comes before translation in the dictionary. Transcription happens first, translation happens second. And don't forget there's another molecule involved, transfer RNA. A tRNA is what brings the amino acids to the ribosome, and it's complementary to the messenger RNA. So if I practiced right here, I'm going to go from DNA. This is my strand of DNA that has the information for a gene. I'm going to take it over to RNA. So a T pairs with an A with a U, remember, C with a G, C with a G, G with a C, C with a G. Adenine will pair with uracil, thymine will pair with adenine, and G with C. We'll break them into some groups of 3. Those are our codons, and here's our codon chart. So I can see that AUG is right here, and that's methionine. It's also called start. And GCG, here's my G, C, G is alanine, and UAC, here's my U, my A, and my C. I see that that codes for tyrosine, so that would be a very short amino acid chain. We can examine the relatedness of species by studying DNA. If I wanted to compare species 1 to species 2, I can simply look for similarities in the DNA. These 3 DNA nucleotides are the same. These 2 are the same, but here is one difference between species 1 and species 2. The remainder are identical. So I would say these species are very, very, very similar, just based on a very tiny, tiny piece of DNA. If I wanted to compare species 1, the same one at the top, with a different species, the T's are the same, the A's, the C's, the G's, but here's a difference, here's a difference, here's a difference, and here's a difference. There are 4 differences between species 1 and species 3. There's only 1 difference between 1 and 2, so species 1 and 2 are much more closely related. If we look at natural selection, Charles Darwin went on a 5-year voyage of fossil collecting, observations, drawings, and a lot of thinking and a lot of writing. And basically he came up with this theory of natural selection, that there is competition amongst organisms, there is advantageous traits that some organisms have, and over time those advantageous traits allow an organism to survive, to reproduce, and then the traits become more common in future generations. So let's take a look at the idea of adaptations. And adaptations we can see in white-tailed deer, which are common here in Maryland. If I looked at a white-tailed deer in the wintertime, I would notice that its coat allows it to blend in well with the background, whereas in the summertime they have a slightly different colored coat that allows them to blend in well. And even the newborn fawns have a spotted coat that helps them blend in with little bits of sunshine filtering in through the trees that form patches on the grass. They also have almost no smell, so a predator rarely ever finds a fawn. And if we look at the bottom picture, deer have an amazing jumping ability. This helps them elude predators. A deer can clear obstacle as high as 6 meters pretty easily, even higher if they're running. Gives them a wonderful advantage over some organism trying to capture them. Evidence of evolution. Fossils provide wonderful evidence. So here's a little diagram of the history of horses as told by fossils. 55 million years ago, horses had toes. They were really walking on toes. The earth was much wetter then, and that spread out their surface area of their feet and allowed them to move. They were much smaller animals. Modern day horses evolved in the desert, and now they're walking on a single hoof, which is really not much bigger than the bottom of the surface of your palm, and allows them to run very, very, very fast on dry ground. Another great piece of evidence of evolution happened to be homologous structures. So we're looking at the forearms of 4 different mammals, and we might notice that they all have one upper arm bone. They have 2 bones in their forearms, and they have carpal bones, and then they have digits. All of these are very similar structures, but they're used for different functions. Bats stretch out some thin skin across this part of their body, allows them to fly. Whales have this modified to be flippers, and humans, of course, have theirs for picking things up, throwing things, that sort of thing, and cats would be using theirs for walking. But the gold standard of evidence of evolution we can find in molecules. DNA, RNA, and proteins offer us an abundant amount of information about who is more closely related to whom. The more similar the DNA, the closer you will be together on these diagrams. These diagrams are called cladograms, and you can use traits or you can use molecules. But the more closely you are to another line on here, the more closely related you are. And there's a common ancestor to all of them at the base, if you want to think of this as a family tree. Similar information over here, we're comparing with DNA, and we can see how similar organisms are based on how close they are on the cladogram. I want to finish up with a little bit about classification. So if we are thinking plants, what characteristics do all plants have? They have cell walls, they are multicellular, and they can make their own food. If we thought about fungi, well, they're different from plants in that fungi can't make their own food. They are all heterotrophs. The only thing they really have in common, they're eukaryotic. They have a cell wall as plants do, but it's made out of a different molecule. So fungi become their own kingdom. Animals, of course, are all multicellular, and they're all heterotrophs. But what separates us from fungi is we have no cell wall. The fact we have no cell wall, it allows us freer movement. And bacteria are our prokaryotes. Bacteria, remember, have cell walls, but unlike eukaryotes, they are all unicellular. Some are capable of making their own food, others are heterotrophs. But the fact that you have cell walls and they are all unicellular is why they are classified the way that they are. And finally, we have the junk drawer of life, the protists. Basically, if you're not a bacterium, not a fungus, not an animal, not a plant, then you happen to be a protist. And with that, I'm going to say make sure you go to class and ask a lot of questions.